name is Jason Baer. I'm a PhD student in the Rower Laboratory, and we are studying some interesting mechanisms behind how a coral reef recovers after disturbance. And so I want to start by introducing two different types of reefs to you guys. And the first is a reef dominated by coral, and this is my best approximation of a coral here. And the second is a reef dominated by fleshy algae, which we see here, or macroalgae. Now, a reef dominated by coral, corals create this incredible three-dimensional structure. And that three-dimensional structure does a few things for us. It provides us with food in the form of fish, because it provides this habitat that supports all kinds of different organisms that also support coral health. And it also helps slow down wave action to the shoreline and actually protect our coasts. Now, on the other hand, this on a reef dominated by fleshy algae, this three-dimensional structure that's created by corals erodes away. And so what we end up losing is a lot of the macroorganism life, like the fish, like the invertebrates here, that we would normally be seeing provide good things for coral health. And instead, what replaces them is a lot of bacteria. Now, algae release these compounds called dissolved organic carbon, or DOC. And this DOC supports the growth of these really large, abundant, and inefficient bacterial communities here. Now, these bacterial communities cause a lot of coral disease, and they draw down oxygen at the sea floor. And in doing so, they actually cause more coral death and leave more space for algae. So what we see here is something that we call a feedback loop. And this feedback loop is really driven by this increased production or growth of algae, which fuels the production of dissolved organic carbon, which fuels the growth of microbes, which causes coral disease and death, which leaves more space for algae. So we call this feedback loop the D-dam feedback loop. And what we think is that this feedback loop is more or less how coral reefs are degrading all across the world. So what we really see is this shift from coral-dominated to algal-dominated reefs worldwide. And what's of interest to us is figuring out how we can break this feedback loop in order to promote coral recovery and get this shift back to coral-dominated states. So to do this, one of the first things that we recognized is that at the seafloor, this high bacterial numbers and an increased DOC actually make this seafloor a very challenging place for coral reefs to be able to recover, for corals to live. So to help this ecosystem try and recover, one of the things we've been working on is actually moving a reef community up into the water column. Now, in doing this, we think we can get more flow, more oxygen, more light, and we can distance these reef communities away from things like algae, away from sedimentation, and other negative effects that these corals experience on the seafloor. So to do this, to move a reef community up into the water column, we had to do two things. First of all, we had to have a way of actually moving a reef community onto a confined space, so of building a reef. Now to do this, we're using a tool called ARMS units, or autonomous reef monitoring structures. And these things were developed in the past few years by people within our lab as well as at the Smithsonian. And what these structures do is they're basically stacks of PVC plates with space in between. And it turns out when we put these out on reefs, they passively accrete all kinds of species that we see on the inside of a reef community. And so they end up getting fouled by all of these organisms, bryozoans, tunicates, sponges, crabs, other small invertebrates, that it turns out are really, really crucial to maintaining coral health. So what these ARMS units do is they actually collect up to about 80% of all coral reef biodiversity. which gives us a tool that we can use to collect and move a lot of these critical components of the reef. So our plan is, and what we're working on now, is that we take these arms units as well as corals from the reef, and we add them on to these structures that we've built called coral reef arcs. Which then we can move up into the water column and actually support these reef communities. And so how coral arcs work is we basically attach them to the seafloor here, and then they're tethered up by a buoy here. So they're actually raised off of the seafloor, and they're hanging out in the midwater here. Once again, where we have better flow conditions, better oxygen, better light, and things like this. So what we're working on now is deploying about 10 of these structures in Curacao, 2 in Puerto Rico, and 5 in Madagascar. And in each of these different places, we'll be looking at different effects of coral arcs on the surrounding ecosystem. For example, in Madagascar, we're using these structural communities to try and recruit fish. So we're looking at the potential of using arcs for fisheries. In Curacao, we're looking at using these things to slow down wave action, so to promote that, that service that we know is really, really important. So three-dimensional structure for wave action. And then in Puerto Rico, we're using these structures to try and restore a coral reef ecosystem. And so by building this reef community on arcs, what we'd like to do is use these to actually repopulate degraded systems. And importantly, these arcs communities, we think, will be able to support existing efforts as well in the coral restoration sphere, such as serving as nurseries that you can take corals from and actually plant them back out onto the reef. And so this is one way that we are trying to both understand the mechanisms that drive coral reef degradation, as well as understand how we might be able to help them recover. And that's what I have for you today. Thank you.